Greetings, class. This is the last part of the module for this semester. And a number of questions appearing in your next test are coming from this module. I have given a scope that covers areas where a number of questions will be drawn. I want at this point to have you recognize three important parameters. One is the selection differential. It refers to what is done in the base population. The base population is this one here. That's where you will choose your best parents out of a base population to cross. That is where we talk about the selection differential. The selection response R, which has to do with the genetic gain, is what is represented in this distribution here. And of course, as you shall see, when you have done the crossing, you grow them out here, then you undertake the process of selection. And therefore, when you are doing the selection in the offspring population, it is represented by these three formulae, which are extremely important for you to note. The formula one here, which is the genetic gain over time, as you can see, the R sub T, is useful and is given here with the indication of the things you must do during the evaluation to achieve this prediction of the genetic gain over time. They are here, 4 of them. This same formula is expressed here in terms of the delta G sub here, which is expressed as is shown here, and these symbols actually represent the same thing as here. Remember, the years per cycle are simply the generation interval. F2 is a complete generation interval. F3 is a complete generation interval. F4 is a complete generation interval. And then we have this particular one here, which expresses the genetic gain as heritability. And you can now see the issue of heritability coming in, multiplied by the selection. And the selection here involves these components, as you can see, and you can decompose the heritability here into the additive genetic variance, where in the denominator you subtract all these unwanted things, which then you have this proportion expressed on the sum of these things that are here, and you can calculate the genetic gain. Now, we need to help in a situation here to look at some of the selection methods. We have a recurrent selection. Something that recurs is what comes, time and again. It can involve a recurrent selection within population, within a population that is intra-population recurrent selection. You can select, between or among populations, and that is called the combining ability recurrent selection. Then the second one is the selective breeding, which involves the aspects that I have indicated here, the selection breeding. And as we can see, there are a few things under the selective breeding when it comes to selfing of crops. But here, the third one is the mutation breeding. Then we have the polyploid breeding. Then we have the tissue culture breeding. Then we have the transgenic breeding. Now, remember, we have the standard breeding, sometimes called the phenotypic selection. And then we have the genomic, which is more modern, more contemporary. And the bottom line, the underlying foundation of either the standard breeding or the genomic selection is making crosses, making crosses, making crosses, making crosses. In the standard breeding, you can see that if you make crosses, you generate inbreeds, then you isolate the lines, and then you have small-scale trial for evaluation, and you can push those small-scale trial evaluations to large scale. And when you are doing a recurrent one within the population, intra-population, then you make crosses again. And that can take from 5 to 7 years just doing the recurrent within a standard breeding strategy. And then you can sell or release the varieties, and that takes 10 to 12 years. So if you take 10 to 12 years plus the recurrent that is from 5 to 7 years, you're talking about 19 years plus. Takes a long time. But in the genomic selection one, you make the crosses and you can double the haploids. And when you double the haploids, it takes about 2 years. And then you can move to small-scale trials. In these small-scale trials, you feed the data you are observing into the model here. And then, of course, you can predict the breeding value, the genotype and the value you can predict of the genetic gain. And you can go on and move to large scale. And then you also feed the data in the successive stage to the model. And you can also then predict the value. Then as you do this, remember, you can also feed the data from the others. This is software based. You feed the data from other efforts and you can predict the breeding value or the genetic gain. And then of course, when you have predicted the genetic gain and it is maximized, then you can sell or release the cultivars. So you see, here you have taken 2 years plus 4 years, and it is possible to come to releasing a new variety between 6 to 7 years, or sometimes to 12 years maximum. Now, let's change our thinking to this slide here. In this slide here, I will want you to put in your mind the stages that we are going to be talking about, the seed being stage 1, the planting seed as stage 1, and then going to when it is now inside, it is germinating inside, that is a germinant, if you want to call it, as stage 2. And then, of course, you have the stage 3, which is an emergence, and then you have a young seedling, which becomes an adult seedling at stage 5, vegetative. It is no longer seedling, but a vegetative plant. And then we have stage 6. Stage 6, stage 6, stage 6. Just remember, stage 6 is where the sex or the sexing happens. Sexing at stage 6. Remember, sex at 6. Sex at 6. That's where a plant breeder comes in to do a crossing. Once this crossing is done, then this seed is allowed to come to stage 7, where you have the parent, which was a particular generation, having new baby generation born in this. At the sex 6 stage, remember, you selected the parents to cross. You selected the parents to cross. You've seen them as they are growing. You evaluate those parents. Remember the chronological order of evaluation as you evaluate. It's just not one plant evaluating, but several of them. Because it is several of these, you'll choose the best parents to cross. Because when you now have this, the process is you are going to select the parents, and of course, you matriculate the parents to cross. You bag them, you tag them, you pollinate them, then you wait for the seed to mature, and you can harvest the seed as the next generation. Let's switch our attention to this integrated diagram. It is rather overwhelming, but in what I have circled here, you will see that it really refers to the breeding methods that we talked about, where again, as you see, we have the tissue culture, we have the interpopulation we're talking about, and we have the interpopulation we also talked about, and then we have these other ones, evaluating for developing combining ability, recurrent selection by a test. And then we have these other ones, when you make crosses, single seed descent, you have this one, pedigree, you have the bulk, you have the back cross, you have the clone, and you have the mutation breeding. And then you get to the processes that will give us certain things that go to the agribusiness world as released variety, foundation seed, registered seed, certified seed, and the grower production seed that a farmer buys from the stockist. So we have this short video which tells you about the sex at 6. The sex at 6.
Ever heard of a pluot? It's a cross between a plum and an apricot. What about a tangelo? That's a cross between a tangerine and a grapefruit. Those are the results of seed breeding. It's kind of like how we got the Labradoodle, or Liger. You cross two breeds to make a new one with the best traits of each. But how does it work? Let's say you wanted to make a consistently blue tomato that could be sold to grocery stores and chefs nationwide. And let's assume blue tomatoes already occur in some wild tomatoes in nature, but not consistently enough to sell to the masses. That's where seed breeding comes in. Most conventional seed breeding takes place in greenhouses to have better control of the environment. Breeders would then plant seeds from those wild tomatoes, the mostly blue ones, and seeds from a successful domesticated tomato, a variety proven to grow well in the area, but aren't naturally blue. When those tomato seeds begin to flower, a plant breeder would remove the pollen from one flower and place it in the flower of the other variety. This resulting batch of tomatoes born is called F1 in the seed breeding world. Breeders will then plant the F1 seeds from the especially blue tomatoes, and those tomatoes are pollinated the natural way, without human interference. What grows is called F2, the second generation. Breeders will pick the bluest tomatoes of that batch and plant them. They continue to manually select and grow seeds from the bluest tomato plants until they see consistency in color and other desirable traits like disease resistance. In plant breeding, it can take dozens or even hundreds of generations to get the consistency and quality you're looking for. So finally, however many generations later, a truly blue tomato is born and is a hit with chefs and farmers across the country.
Great. Fantastic. Now, this is just a small question with an answer to help you see where we are going when you're talking about the importance of sex at 6. Sex at 6 stage. Sex at 6. And this is also to inform you. It's just something you should know. So if you have a donor parent, remember, you need about 250 females to pollinate the donor on, so that in your F1 seed, you can expect to harvest about 1 kilogram of seed. Now, there are ways a breeder can manage handling crossing of many plants in a single operation. One of them, if it is maize, for example, you can, or other plants, you can emasculate or remove the anthers of the unwanted plants, or if you are crossing, you can make certain plants to be males and certain plants to be females, and you allow the pollination to go on. Or you can manually pollinate by actually transferring the pollen from the donor to the receptor, the receiving parent. Now, let's just go back again to the various methods that are common for both self and cross-pollinated. Remember again our big figure, the big figure with a lot of the details of the various breeding methods. And here, some of these methods, backcross method, may involve sex at 6. Polyploid method breeding may not involve sex at 6 because polyploid is a different game altogether. Mutation breeding may not involve sex at 6. Transgenic breeding may not involve sex at 6 unless you have now produced those plants and you want to go further and sell for a reason. But the ones which may not involve sex at 6 may be where you may need to sort of now select what you want as the resulting generation from. Now, for inbreeding species are those that are self-pollinated. The techniques or schemes of selection are these 7 that are actually indicated here. You can read them yourself. I will send the PDF notes to go with this presentation. But let's get to this example here, which is very important. Here, of course, this is double S. You have a parent population, a base population, where you select a good parent and you cross a good parent with a good parent. And when you cross them, you have selected them from the base population. Remember again, therefore, the selection differential is important when it comes to the base population here. And then when you have crossed the best 2 parents you have selected, they will give populations that are very many plants, hundreds or even thousands of plants in a given area in a nursery. This is where then you can look at the selection response. Remember the formula that we used before. So the important message here is this information in this graph and this information in this graph. Please remember it. Remember it because you might also see that as a possibility in any subsequent exam or test, or just for the sake of remembering. Now, you can see, for example, in the wild mustard plant, that is an example of what we are calling the CWR, the crop wild resources. And you can see just the structures from the same plant, the wild mustard plant. It has pioneered into developing them to these various products. And they are really very monumental. And you can see that each of the structures from here have the allele determining traits. I mean, the trait determining alleles, where they have resulted in the cabbage from just this here. You can see the terminal leaf bud with the various genes, alleles that contributed to the development of a cabbage. And this coming from the leaves, the trait determining alleles giving rise to a kale. And you can see kale is sold at the Rundu market in the Kavango area. And then you have the bit coming, the trait determining alleles for this structure, giving rise to cauliflower and so on and so forth. So each structure may have certain trait-determining alleles that plant breeders can take advantage of, right from the tuber structure or root structure up to the seed structure. And these are the results of the products from the ultimate target and the ultimate or the final products to this day from the selection response. The selection response. Right. As we move on, we want to change our mindset as we go along from one area to another. I have here a summary of the most common selection schemes. We have the issue of when for mass, pure line when it should be done, pedigree when it is done, back growth when it is done, and marker assisted or genomic selection when it is done. The summary goes further on mass selection when it is done, pure line, pedigree, backcross, not when, but how. It's a summary. And the rest here, I will not really go into this detail. It summarizes, and it is best for you to look at the differences between the self-fertilized on the left and the open pollinated on the right and see the differences. This goes on in also looking at the left for self-pollinated and on the right, the open pollinated. It is simply expanding the mass selection method. And it is sometimes modified from the original. And remember, it is where you are talking, where it is talking about the cell and the mass selection. Right. So you going on to look at these differences because here the mass selection, you are dealing with individual plant selection method. All right. So when you come to the breeding methods in cross-pollinated crops, you then have those two: population improvement and the hybrid and synthetic varieties on the right side. And it goes on to indicate how you go on doing this. Look at the slides. And again, this is simply a detail you can read. I am going to send you a PDF to go together with this. I do not have to repeat. And then we have the reciprocal recurrent selection that is also shown here schematically. Please study it carefully. And it just shows a number of things regarding the reciprocal recurrent selection. Just look at it again closely, which I cannot go, I cannot really explain longer in the online voice presentation. And the details go on in a little more detail that you should be looking at. And then we have the populations for progeny selection. Again, they are shown in this detail you should look at and get to understand them on your own up to this level that involves these bits that you need to study from this. Thank you.